Hello everyone, my name is Jorna. I'm a PhD student with Dr. Byron Adams at Brigham Young University, and today I want to tell a story about community assembly in a high polar desert and some implications that might have for inter-trophic interactions in this ecosystem. So we work in the dry valleys. As a quick recap, they are located in this red box, which is roughly where Victoria Land is in Antarctica. And there are these valleys here called the McMurdo Dry Valleys that run from further inland to the coast and are blocked off by the Transantarctic Mountains, which keeps them free from most glacier ice and any big ice mass to develop. And so the valley floor looks mostly like this. We call this desert pavement, so it's mostly rocky and sandy, and it might have a snowpack or a bit of wet soil every now and then. When we talk about these ecosystems, it's really a story of nematodes and microbes. So I always like to show this picture to really illustrate that bacteria and nematodes kind of live on the same scale, right? So they inhabit the same world. You can see a nematode here, a nematode's face, or head, I should say, covered with bacterial cells. So yeah, really operating at the same kind of scales. We don't have this specific nematode in the dry valleys, but we have other ones, such as this Scotnema lindsay. And so that's really the ecosystem we're dealing with. We don't have any plants or higher invertebrates or vertebrates, so we really only have nematodes feeding on these microbial species. And so kind of the whole range of ecosystem function that you would expect in other systems, whether they're terrestrial or marine, are kind of exhibited just by these two levels in the food web here. And so to simplify even further, we were curious about how communities in these systems assemble. So for instance, after disturbance, let's say glaciation or opening up of new habitats because of climatic reasons, how do communities assemble over time, especially nematodes? And then if we do an experiment such as this, what can that teach us about how these species interact, how they functionally relate to each other in the ecosystem? So to do this, we set up a very simple experiment. We basically took soil that we knew was already a viable habitat that was inhabited by nematode species such as the Scotnema lindsay, and we sterilized them. So we put them in an autoclave, we killed all life in these systems, and then re-incubated them in the field in these nylon mesh bags, and basically tracked over time what happened to these communities. So this is what the data look like. So we did this in 8 replicates, and I'm going to show you mostly the nematode counts. So this is the number of nematodes we found in 100 grams of soil over time. So on the y-axis, number of nematodes, and then we're going to go forward in time throughout this plot. So in red, you can see the control plots. So before we did any work, we sampled these plots. We see about 100 nematodes per 100 grams of soil in these samples. And then we sampled them after sterilization, right? So the first little blue bar is right after sterilization. As expected, everything should be dead. The second blue bar is a year later, still no nematodes. And then once again, 4 years later, we still didn't find any live worms in these soils. Curiously, what we did find is the data is a little bit harder to show here, but we did some soil respiration measurements. So we actually know that bacteria were producing carbon dioxide in these soils already in the first year and especially after 4 years. So we know there's bacteria present. We just don't know exactly what they are. And then after 15 years, so last year, we sampled these plots finally. We see again in red control plots. So we sampled new control plots after 15 years to see if those had changed. And then we see the blue bar are sterilized plots finally have the first influx of nematode worms. So we have a few out of our 8 samples, I think it was 4 of them, that now have worms in them, but it's still a very low population number compared to a healthy sample. So the real question now is obviously what is happening during these first years and what changed in the last year. So the idea here is to sequence the metagenomic profile of these communities, looking mostly at the bacteria, so to see which bacteria arrive first, how they change over time, and which bacteria especially are there once nematodes finally arrive, and how those are different between soils with and without nematodes, and different from the control plots. So what could that tell us? Well, it told us already that nematodes are very slow to recolonize available habitat in the system. So 15 years is a pretty long time for them to become reestablished, even in very small numbers. And this is probably due to these very key bacterial guilds that are very relevant for their establishment, that have functions that they require in soils. And so doing this metagenomic sequencing can really inform us which functions are there to facilitate nematode survival and colonization of these habitats. And so I'm very excited to find out what these results will show us and hopefully share them with you in the future.